So what I'm going to talk about today, I'm going to spend the first 10 or 15 minutes here giving a general introduction to the setup of the course, what we're going to do, what it is that you have to do, and what you don't have to do. And then I'm gradually going to go more and more into the actual biology. I'm going to cover the literature too, and while we're at it, I might as well pass around the lecture notes. I'll come back to that. I might continue printing those. It depends a bit. If I realize that half of the team is away and watching it on YouTube, again, perfectly fine. But then it's a horrible waste of tree for me to print 30 copies of the lecture notes every time. So if it fluctuates a lot, I might just ask you to download the lecture notes as PDFs. I know that some of you like to use iPads and take notes, so that I have both handout versions and the full slide versions if you want to download them and notes. But before we get there, I'm going to spend a little bit of time introducing you to biophysics. Biophysics is a very broad topic. And I'm not even sure whether there's one true definition. But the idea of biophysics is to somehow connect, on the one hand, biology. That's the bio part, obviously. Biology is the real wet lab biology, as you see up on the left. Zebrafish is traditionally wet, right? And that's not at all. Historically, it hasn't been a quantitative subject. That has changed a lot the last 50 years, and in particular the last decade, I would say. Because first we're getting so much more information, the whole sequencing and everything I'm going to touch on in a few minutes, but also that we're gradually being able, as we're understanding more and more on the molecular level here, we're able to not just go top down, but increasingly also bottom up. So zebrafish might seem like if you're a biologist, it's probably a very fun topic, but if you're a physicist, it might seem completely pointless to study zebrafish, right? On the other hand, there are quite a few people who have been able to do models. This is based on computer simulations from a colleague of mine, actually, Petros Koumoutsakis in Switzerland, where they've been studying fish and how fish swim together. And the reason why fish swim together, it actually turns out that it's energetically more favorable. And they can even do computational fluid dynamics simulation now based entirely on physics and calculate roughly how much energy they're saving. And that helps you to actually predict not just patterns, but a whole lot of this type of research is then being used to things like patterns in car flow, or for instance, if you have trucks, should you let the trucks drive together to, in similar ways, save on the air resistance. Now, the reason why we're starting with zebrafish is there is actually a point to this. It might seem completely pointless to study. Researchers are not necessarily enemies of pointless things. So what you can do, if you have zebrafish in a small aquarium, you can put a camera on top of the aquarium. And as you have this camera, you can start recording the individual zebrafish, and both as a function of time and location in the aquarium, you can then start to plot this in 3 dimensions and see how do the fish swim. And if you have any numerical way of characterizing how the fish swim, you can then check, so what happens if you now, say, add ethanol or nicotine or caffeine or anything? And then you start to see differences here. And there are 2 important things here. The first thing is that this is noisy. It's insanely noisy. So what are your backgrounds? Are both of you physicists or a few chemists? Physics? Okay, great. Some of the things will be repetition for you who are physicists, and conversely, some of the things are going to be repetition for those who are chemists. You can certainly characterize this, and compared to classical physics, the challenge here is frequently the noise. And if you do this experiment a second time, you're going to get, not just different results, but the results might be so different that you can't even reproduce it the second time. And that's a particular challenge with biology. It's far more varying than, say, trying to measure Planck's constant or something. So why do you think you do this? Apart from the fact that you might, 50 years ago, people might have been interested in, say, studying animal behavior or something. Today, it's much more concrete. We even have facilities for this within SciLife Lab. So what you're doing with zebrafish, you're actually using this as screens. For instance, if you want to test the effect of new drugs, chemicals, what dose, assuming that caffeine was a new drug, what dose of this drug should you administer until you start seeing changes in behavior in the brain or something? That's probably an experiment you don't want to start doing in humans, right? Zebrafish, on the other hand, perfectly fine. You hardly need you do need a permit for it, but it's not a very difficult permit to get. So this way you can try, it's physics in the sense that we're measuring the location of zebrafish with camera or sensors, right? It's physics in the sense that you're analyzing the patterns. You're going to need physics to describe, is this nicotine pattern significantly different from the caffeine pattern? Yes, you can probably say that, they look completely different. But what does completely different mean? Does it mean that it's 1 chance in 10 that it would happen? I would not be comfortable administering that drug to myself. 1 chance in 1,000, 1 chance in 1,000,000. And then we're getting back to the statistics. So a whole lot in biology has to do with statistics. And although I'm going to spend quite a bit of time today talking about biology and structure, on Friday we're going to get back to physics. And the more we get back into physics, the more we're going to bring in statistical physics. But what I love with this course is it's a completely different way of bringing in statistical physics than you would normally do with, say, statistical thermodynamics. The power of this way of reasoning, combining biology with physics, is that you can also start to think of it in terms of scales, in length or time, space or time. Sorry, this is a bit of a faint image, but on the top left you have a tree. And that's probably a scale of tens of meters. If you then zoom in on, say, a leaf or something, you might have a scale of a centimeter or a few centimeters. Inside this leaf, you're going to have, say, a cell, which might start becoming a scale of a millimeter or something. And then you have components of the cell, which starts to be a scale of 10 micrometers or something. Inside those components of cells, eventually you're going to get into very tiny organelles in the cell. And eventually somewhere in there, and this is a model, not a structure, you're going to see a small protein, which might be part of the photosynthetic reaction cycle or something. And this component is partly what explains why the tree is green, but also what explains the entire respiratory cycle and everything. Now the challenge here, how on earth do you describe a tree at the same time as you try to describe a molecule, right? A tree has a lifetime of maybe 100 years. The cycles we're going to need to do when we describe these molecules and how things are moving in the molecule is going to be maybe a millisecond. And that's insanely difficult. And I'm sorry to break it to you, but we don't really have any good ways or mathematical formalism or anything of treating all this at the same time. So that means that we end up both with the reductionism and try to simplify. We can certainly, historically, this is the way we've always done it. You tend to go top down, usually with observation. The classical way of doing this is a microscope. And as you have better and better microscopes, we are able to drill further and further down. What has happened, though, in particular the last 40 years, is that we've become increasingly better at studying things on the molecular level, partly based on physics and structure, I'll come back to that, but also partly because we've gotten so good at understanding what the structure is that you can use physics and model it so that, hey, if we know that that's the structure, can you predict what color this component would have? We probably can, because we can calculate what wavelengths of light it would absorb, right? And then you start to introduce a bottom-up approach instead. If we know what the components are, can I predict how the entire organism would work? And that's kind of similar to the zebrafish, that I want to try a chemical on a zebrafish so I understand the influence this chemical has so that I can use this drug in a patient, which is a much more complicated system. And, well, I'm not sure whether this is an ideal illustration, but you could even plot this two-dimensionally, where you have some sort of scales from microscopic things on micrometers or even nanometers up to macro scales with humans, or even, you could even think in terms of population dynamics. We're not going to talk about that, but the entire Earth is kind of an ecosystem, right? So you have tens of thousands of kilometers. And same thing in times, you have things ranging from nano or even picoseconds up to, well, if you think of the population, you're talking about 4.3 billion years, right? Evolution, which is somewhat difficult. You usually don't go beyond the Earth in biophysics, not yet at least. What we're going to focus most of our attention on in this course is this part of the spectrum, understanding molecules. And it's not because the other things are not at all interesting. Actually, I'm going to try to connect things up. But this is really where physics is starting to play a pivotal role. We're completely changing the way we can interact with things. And I would really encourage you to think of this in terms of the top-down observation versus bottom-up modeling. Both are important, but we will probably put a bit of our attention here too, except for today, that we're going to do more observation. There are a bunch of fun systems that I'm going to keep touching upon, and don't worry too much the first time I bring something up if you don't understand it fully, because if it is important, I will be coming back to it again and again and again. And what I both love and what can be difficult in this course is this ping-ponging back between biology and physics. And that frequently requires me to first introduce something conceptually. Let's pick an example. These are the channels that are pumping different types of ions in and out of your cell. This is a super complicated topic because it involves your entire nervous system. This is the engine in your nervous system. We're using ATP for it, but we also have to understand what a membrane looks like. You have to understand what the physics look like. There is no way I could introduce all those things at the same time. So that means in this case, and we're going to talk about this later on in the course, we would first say what does it do biologically and not worry too much about the details. Then start to go into the physics and what is the physical part of this process. Once you understand the low-level physics, I can take a step back to the biology and I started to say, okay, so how will the channel based on that physics work? Once I know how the channel works, I can start to think in terms of a higher level physics. What would this mean for all the different conformations the channel could be in the energy landscape, and how could the channel change its behavior and eventually bring that back to biology again? So the simplest way of thinking this, channels are just holes. Pumps, on the other hand, they can transport ions in the opposite direction. That is against, in this case, a gradient of the ion. To do that, they would require energy. And in this course, we haven't yet touched upon where you get that energy from, but we'll come back to that later. There are, of course, structures on these channels that too we will go through. The energy you see somewhere here, that it's catalyzed by, say, ATP. It's a small molecule. Again, this too we're going to cover later. And the reason why all this machinery works in the first place, or rather, the reason why we can understand how the machinery works, is that since roughly 20 years, we have very detailed, we have not just molecular models, we have very detailed structures of this entire pump complex. We know we know roughly where every single small atom is sitting here. These are amazing machines. Even after, Jesus, I was about to say 20, but I probably had 30 years in this field by now. Even after these 25, 30 years, I'm still in shock and awe that these machines work. I can't understand it. Well, we can understand it, but that nature has actually evolved to this. And as a physicist, the amazing thing with this 4.3 billion years of trial and error evolution is still a miracle. There are a bunch of other proteins that we're going to touch in. I will just give you that final example before we get back. G protein coupled receptors. When I was roughly your age and studied undergraduate engineering physics and biophysics in Lund, and then as a PhD student at KTH in the late 1990s, we didn't have these structures yet. This is called GPCR, which is a G protein coupled receptor, which sounds fairly boring, but this is probably the most central component in all signaling. When cells need to talk to each other, when you're binding something that one cell is going to tell another cell to start a process or something, there are dozens, if not hundreds, of these different genes in your body. So they're literally the entire communications network. And until 20 years ago, until 50 years ago, we just know, yeah, it exists, and it must be a protein, but it was just a black box. We had no real idea what it was like. We knew roughly that it's sitting in a membrane, which is obvious, because if you're going to need to send a signal from the outside of a cell to the inside, we need to get across the membrane. And at that point in time, people were fairly negative, and I remember sitting through a bunch of seminars where people said that, look, we've sunk $2 billion in this field. The likelihood that we will ever see a structure of these proteins is zero. Until people found a structure of it. And now there are probably 50 structures known. So that's the other cool thing compared to physics. As much as I love physics, traditional physics evolves fairly slow. Biophysics evolves exceptionally rapidly. Probably half the stuff I'm going to tell you in this course was not known when they printed the first edition of the book we're using. Which is, again, a challenge because it's difficult to find good literature, but the cool thing, things happen in this field every single year. The reason why we have a structure of this, I'm going to come back to that too, is that you're using X-ray crystallography. Roughly the same way that we first determined the structure of, say, normal salt, sodium chloride. But I'm not sure about you, but this is slightly few more atoms than sodium chloride. On the other, when we have a structure, you can start to do any type of modeling you can imagine. So some almost 40 years ago, people realized that this structure, it's just a bunch of atoms. They will obey the laws of physics. And if they obey the laws of physics, well, you could in theory put them in a, well, now we can, not just in theory, but in practice. We can put them in a computer and then determine how this G protein coupled receptor binds a small molecule ligand. And you see that it got really stuck there. And now that the molecule is stuck, this is changing the structure of this entire protein. And when the structure of the entire protein has changed, what this will lead to is then a chain effect on the inside that will cause the signal to propagate on the inside of the cell. Again, we're going to come back to this several times in the course. And that leads to the extra complication here that I don't think either of these books do, that the last 10 years or even 15, biophysics has increasingly become computational. not in contrast to experiments. We're using both computers, and computers are increasingly our new experimental tool. We use both experimental observation, we're collecting tons of data, and then we're using computers to make sense of it. So what I showed you here was run on a very large supercomputing called Anton in New York. It's the one on the top right there. But there are a bunch of these machines. All of these machines have at one point in time been the largest in the world. This is the new Chinese machine. They're basically, people are investing billions of dollars to build larger and larger machines to use essentially as computational microscopes, because we can use computers to study things on scales and with detail that is completely impossible in the lab. Again, this was science fiction when I started my PhD in these topics, but today, any modern drug you design now has partly been designed in a computer. It's too inefficient to do it in the lab. So what that allows you to do, you can, for instance, compare the docking, and again, that would basically mean the computer trying to predict how should this small drug bind. And what you see here in purple is how the computer predicted that it would bind, and what you see in gray is the X-ray crystal structure prediction, how it would bind. It's pretty darn accurate. The difference is that determining a new X-ray structure of such a molecule can take months, if not years. The first time it took a decade. Doing this in the computer, you can probably do one in a week. And in the future, people hope to be able to do one in a minute or so. And then you can imagine screening through 1 million drugs in parallel. So the reason why people are interested in that, both our teams and others, is partly understanding physics and interactions, how molecules work in the first scale, so that again, these are just molecules. There is no special life essence or anything. They're just stupid molecules in a way, but when they are these large and complex, they start to behave in different ways than water or carbon dioxide, which would be the traditional molecules we're used to. And this is exceptionally important, particularly in the pharmaceutical industry. I don't know how many billions of dollars are in this slide, but these are basically companies and the trademarks, generic names of drugs targeting just G protein coupled receptors. And I'm going to come back to this later on in the course, but we're having the average revenue for these blockbuster drugs can easily go in the tens of billions of dollars today. So it's exceptionally important commercially too, not just from science. I'm going to have one more example before I go into the course specification. We can certainly zoom out. You can go from that scale of molecules to entire cells, starts to look where in the cells things are present. There is actually a lot of work on that going on out at SciLife Lab that I might tell you a little bit about. We can start to look where things are in different types of tissues, organs. Are there differences if a patient, say, has a cancer tumor? Are there going to be differences in what proteins are expressed where in the body or how the proteins behave? In general, the answer to that question is yes. So we would maybe like to design a particular drug that would just target some proteins that are expressed too much or too little, or that have changed in a way so that they don't behave the way they normally would. On the other hand, you could also go the opposite way. You could start with something from the outside. Let's say that I have a traditional drug that I would like to deliver. And deliver is the way that I would like to get this into your cell, because if the drug is just on the outside, it doesn't help a whole lot. The problem is that your bodies are designed to protect the cells from the outside. That's what the skin and everything does. And you could certainly try to inject a drug, but drugs that you inject, they're virtually never successful. It doesn't really work. Well, it works, but it's expensive. You need to go to the doctor 3 times a week, and you need to take injections 3 times a week. If you're a diabetic, you have to do it, but again, it's difficult, and you need a lot of hygienic equipment and everything. So by far the best drug is a drug you can take as a skin patch. And there are contraceptives that can be delivered that way. There are a couple of anti-nausea pills, if you get seasick, for instance. But most drugs can't be delivered that way, because if you zoom in from the outside, what you have here protecting you are parts of your cellular component. This is the lipid bilayer you see up there on the right. And the whole idea with this is to protect things so they can't go into your cells. Here too, you can use computer simulations. We have a bunch of people 3 people in our lab sitting and working on this together with a company, and they're trying to design computational models that really model exactly how do the lipids look like in these upper parts of your skin. And if we know how the lipids look like in the upper parts of the skin, then you kind of understand what are the properties of this barrier? What is it that causes some drugs not to penetrate, but what causes other drugs to penetrate your skin? And if you do that, you can start to build a computational model of this. And when you do that in turn, then you can start to let the computer screen through thousands or tens of thousands or millions of drugs and see, first, what drugs will be able to go through the skin. Those might be the drugs you want to focus on in your experimental trials, right? Second, if you have a drug that you would like to deliver because it's a really good medical drug, could I modify this drug, add a group or subtract a group So I'm able to deliver this in the skin. Same thing here. This could easily run to the millions of dollars and investments, but of course also much better quality of life for the patients. And this is very much based on physics. What is it that determines whether things happen or not? Well, it's partly diffusion, right? That you've probably all seen in physics, and partly concepts like energy, free energy, interactions. So before we know it, already on Friday, we're going to begin drilling down here and start to look how do all these different molecules interact, the electrostatics, van der Waals, bonds in the molecules, and then we're going to start talking more and more about energy for a few lectures before we head back into biology. There are a couple of other proteins that we're probably going to mention simply because they're very important in our research. And again, the beauty of this course is that we can connect things close to research. What you see on the left there is literally the world's smallest machine. It's just 4 helices, it's probably 10 nanometers across, but every time the voltage across your cell changes when you have a nervous signal, for instance, when I'm moving my hand, there are millions of these. The blue helix in the background will move up a bit and cause a chain effect of other channels to open. What you're seeing on the right here is a molecule that's a ligand-gated channel, so that if you're having a glass of alcohol on Friday, the ethanol molecules will bind to this channel and will actually cause the channels to open a bit more than it would normally do, which again will alter your nerve signals. So there are really cool ways that you can fine-tune and interact with the nervous system. These type of channels are also what makes it possible to sedate you with anesthetics. And again, we know what they look like nowadays. So there are a ton of proteins that we're going to keep coming back to. If there is something happening in your body and you don't know what it is that happened, guess that it's a protein doing it. That's in 90% of the cases, that's true. We're going to come back to most of these classes later on in the course, and we're increasingly going to talk about first what they do, but also what you can do here on the right. So on the right, you have an artificial protein, a protein that has been designed in the lab and computers to achieve a specific feature. In this case, artificial photosynthesis. Why would artificial photosynthesis be useful? That would be certainly one part of it, but also converting. What does the photosynthesis do primarily? It converts light into energy, right? And I'm not sure if you're a physicist, you might have read there's been an amazing development in photovoltaics the last decade. You have these new types of much more efficient solar cells. None of the existing solar cells comes close to what happens in a green leaf in nature. So if you could design artificial photosynthesis, they would be way more efficient than the best solar cells you can imagine. And that's an important memento mori, because as smart as we think we are as physicists, we are pretty smart, right? And again, the new photovoltaics are impressive, but nature has had 4 billion years of trial and error to come up with something better. And 4.3 billion years usually beats being smart. So many of these things that are introduced in biology, they're increasingly being used in engineering. Because simply they're more efficient. There are all other ways we try to use bacteria to grow ethanol. And again, carbon dioxide capture is also a very good idea. So biophysics ultimately is very much about explaining phenomena like this. It's more, in some ways it's easier than physics because we're not going to use quite as much hardcore math here. But that occasionally also makes it more difficult, I think, because it's not as well defined as physics. You're going to need to spend more time and everything. Well, okay, that's a biological concept. How do you turn this biological concept into a physical model? So it's increasingly going to be up to us to come up with the equations. Now, those equations we come up with will be much simpler than the traditional physics equations you're used to. But it frequently requires more of you to decide what to model. We're going to do gross, horrible simplifications. And the reason why we need those simplifications is because it's so noisy. Unless you make horrible simplifications, nothing will work. And it's very much about knowing what's a known unknown versus unknown unknowns. That what are the things that we can try to remove from our model to focus on the core properties of it. And increasingly, the great thing is that if we have a simple model, we could of course even put it in a computer. And you're going to use things in computers in this course a lot. I will also try, if you're interested, I'm going to try to take you out to a site visit to SciLife Lab so you can see what happens on the experimental side. Most of what we do is going to be applied to proteins and nucleic acids, but these small structural components, simply because it's, for physics, that's where it all starts. These molecules, they are micro machines, they will move between different states, and the physics and energy of these interactions determines what happens and what doesn't happen. And what I think is really cool, you don't really need, well, you need a bit of physics, you need a bit of common sense, but then you need to think. and you get surprisingly far with that. We can explain not everything, but a pretty darn large fraction of everything that happens in very complex systems. So if we go a little bit into course meta info, time is running quickly here. This is a 50% course, not 100%, as I said at Stockholm University. We will average roughly 2 lectures per week. Some weeks it might be 3. And we have, there are several days per week that are intentionally kept free, partly because you're taking other courses in parallel and partially because we want to give you some time to study. The key word in that sentence is time to study. If you only go to these lectures, you might pass the course, but if you want a high grade in the course, you will have to take some time to do the studies and read up on your own. You will also need to spend some time on these hand-in tasks. There are going to be 3 hand-in tasks in the lab. They are just pass-fail. The first one will have to be handed in roughly 2 weeks from now on. Well, a little bit over 2 weeks. No, sorry, 3 weeks from now. The idea is that we will have roughly 2 weeks for each hand-in task. There's going to be 1 computer lab divided in 2 sessions. You will get separate credits for the hand-in tasks and the lab, and then a written exam at the end. In general, I am a big fan of freedom under responsibility. When I studied engineering physics some 25 years ago in Lund, there were like 6 months with no mandatory moments in the course. I want to give you that freedom too. But remember, freedom comes with responsibility. If you want to go away skiing for 4 weeks, be my guest. In that case, contact us and say that you will not be able to do that computer lab on site. In principle, we're going to okay that. But don't come after those 4 weeks and expect to be able to sit down with us and go through the computer lab. That literally means you get the instructions, you read up on what you need yourself, and you will have to complete a written report instead. Again, responsibility part. Same thing with the hand-in tasks and everything. We don't want to sit the day before the exam and have to correct 30 times 3 hand-in tasks because then we die. So that's why we want to space it out. The other reason we're spacing this out, you're really hurting yourself if you don't do them on time, and that's why we're going to force you to do them on time. Because the first hand-in task, for instance, is very much about the Boltzmann distribution, understanding entropy and everything in a slightly different way than you use in physics. And I deliberately planned that pretty much in parallel with the way we're bringing things up in the lectures, because the hand-in task will force you to approach it in a different way from what we do in the lectures. It will help you to do them on time. So by on time, when we start to describe the hand-in task, don't think that these 2 weeks means you should do it the day before the deadline. Start working on them right away, and then you will hopefully have a much smoother sailing through the entire course. As I mentioned, I will try to record the lectures, but there is no promise whatsoever there. If they're not recorded, they are not recorded. We're virtually always going to be here, and there are some events in FB55, and the computer labs is going to be out in the RB33 room. I'm not going to repeat all the stuff about when things are. All the course information will be in Canvas, and it will be up to date in Canvas. That would have been a really good idea if KTH had actually transitioned to Canvas now after 24 months of time. But KTH thought it would be a great idea to have 3 different places with course information, out of which I might be able to edit some of them sometimes. We will do our best. I've complained to KTH about this multiple times. The Canvas course page, I will make sure that it's up to date. For the other ones, I will do my best. But for instance, yesterday, I couldn't even remove the old schedule from the KTH social site because they think just because I'm a professor and in charge of the course doesn't mean that I have the right to write anything there. Sorry about that. Not too much I can do about it. Full schedule will be there. Did all of you get an email yesterday? Good, because stay there. I don't have your email address there. I can just decide to send an email to you, and then I have no idea whether anything shows up. Feel free to use the forums for anything there. If you can use it to talk to each other. If there are questions you have, we will try to be attentive and respond to the forums. You're more than welcome to send an email to you too. The reason why I suggest not sending an email to me is that I want to spend this time in engaging with you. And if there are 30 of you who send individual emails to me about the same questions, I'm going to respond to that question 30 times. And that means that there will be 29 other questions I don't have time to respond to. But if you said post your question, I will respond to it once, and then I have time to respond to 29 more questions. So if you have a question, there are likely more people that have the same question, and that's why I prefer to do the forums. I will cover virtually all the topics here, and I think the same goes for Lucy and Burke, but do read literature and papers too. I will try to, occasionally I will include some scientific papers. Partly, in some cases they're really useful. It's not that I'm going to interrogate you and ask deep, specific questions about papers in the exam in most cases, But the reason why I hand out papers, they will help you to understand the material. They will help you to understand the book better. And in many cases, they will help you to understand what happened since they wrote the books. There will be other papers that I don't mention so much or that I don't hand out. And in that case, consider it optional reading if you're interested in that aspect. But I'm not going to ask about that on the exam. And since this is an advanced course, a long time ago when we first started this course, there was a beautiful book written by Alexei Finkelstein, Protein Physics. The course was even called Protein Physics the first few years I gave it. And the beautiful part of this was deliberately a book designed as a PhD level course, where each chapter was meant as a lecture. And that doesn't exist on this level. So we used that for several years. The problem is the beauty of this field. The field moves. So suddenly we realized, Jesus, the book is 10 years old. Half of the things I want to tell you, they were discovered after the book was written. Then on top of that, the book went out of print. And then for a few years, people were able to get it on Amazon or private sellers in India or something. And at some point, we had to give up on that and try to see if you could find a PDF copy of the book online, which many people were able to. And then Magnus started to favor another book written by Thomas Nordlund, which is kind of the opposite. This is a usual US textbook. If you like to read lots of text, these books are great. And I say it's a beautiful book, but this goes all the way from the start, and then you're going to spend a lot of time reading here. And I'm not sure about you. What I love with being a physicist, I don't like to read a lot of text. It will probably take you longer to go through this book, because this book doesn't bullshit as much. He goes straight into the equations. So I have to confess, I'm biased. I still love this book, but it depends a little bit what type of material you want to approach. I think you can find both of these on electronic loans from the KTH library. You're more than welcome to have a look on this. I will try to provide literature references to both of them. But again, focus on what we go through in the lectures. Read those concepts in the books too. If there's something we haven't covered at all, we're not going to ask you about that. But the concepts we cover in the lecture are reflected to roughly 80% in both books. And then there are some things where we're either just going to cover in the lectures or cover in the slides. And I realize on the lower level, you want a textbook that you read from the first to the last page. In that case, you can pick one of them. But I will deliberately try to provide reading reference to both. But the course is defined on what we talk about, not the books. Examination, important also. As I said, that we've separated this in two parts. The written exam will be a relatively small part of the course. You will be able to get 5 ECTS credits just by completing the hand-in tasks and completing the lab. And that will basically get you an E grade. No, sorry, you're going to get pass grade. Those two moments are examined separately, so you're going to get pass grades on those. If you specifically would like to show how good you are at this, we will also have a written exam that's divided into 2 pieces. There will be 10 multiple choice questions. That counts for roughly 60% of the requirements. And 60% of those have to be correct. And if you do that, you will get an E. If you want these higher grades, they're going to be 10 essay questions where you have to write a bit. And the idea here is intentionally to try to take some stress off you. If you don't care that much about the grades and everything, it's not too difficult to pass this course if you just follow the course and do all these moments as we're helping you. And again, we will be available to help you, but please don't all come and ask for help with the hand-in task 3 hours before the deadline Sunday evening, because that we can't handle. The hand-in tasks, as mentioned, they're going to be available in KTH Canvas later tonight even, or at least later this week. Same thing there. Don't worry. You have this material available. There's going to be plenty of information there. But it's not going to be like a lab. It's not just going to take 2 hours and you will get click-by-click instructions, do this, do that. You will have to read a bit. You might have to go back to the course lectures and understand what we were talking about. Because again, this is an advanced level course. You expect to search for your own information. And then to make it more modern, that is not at all covered in either of those books. Burke in particular is going to help set up a computer lab where you're going to do proper molecular simulations the way we in our research groups and other people are doing it today. Pure computational modeling and calculating free energies, which is beautiful, very much related to current research. We are flexible. We can handle virtually everything if you contact us before deadlines. Contacting us after deadlines does not make us happy. We have 8 more minutes before the break. So I'm actually going to go through the first few slides on the outlines today. Topic-wise, what I'm going to bring up is mostly basic concepts, a little bit about water, and a bunch of different biomolecules. bit about protein production machinery and the protein structure, and then on Friday I'm going to go into the physics. Before I do that, did you have any questions about the course setup? I'm available in the break too if you have questions about that. So biophysics, the single most important molecule in biophysics is this one, water. This is actually a computer simulation of water. And once in the 1970s, this was science fiction, the type of simulation that got people the Nobel Prize. Today, this is something you can pretty much do on a laptop. So this is liquid water. And you've probably seen ice crystals or something. And liquid water behaves quite differently. In ice crystals, every water molecule participates in 4 hydrogen bonds. It donates 2 hydrogen bonds and it accepts 2. So that means that you have 2 full hydrogen bonds per water molecule. And in liquid water, you have almost the same number, 1.7. So it's not as simple as saying when you're melting ice, you're breaking all the hydrogen bonds. But on the other, we're not really breaking any real bonds either. There is not a single oxygen that's leaving the hydrogens. So this is all a matter of what state is most favorable. Is it most favorable to be in crystal or more favorable to liquid? How are all these molecules interacting? In particular, as you're moving between different states here, this is the finite temperature that causes them to move semi-randomly. And these are things we need to understand. In particular, if you, at say 300 Kelvin, which phase is more favorable for water? It's going to turn out that's not too different from proteins or DNA or anything. So that understanding that if you're studying physics, in particular quantum physics or quantum chemistry, it's usually you're looking at one molecule, right? And then you want to determine what is the structure with a capital S, what is the energy. This is more complicated. I haven't even introduced any quantum yet, but the problem is that we can be in multiple structures, which again is very much a character of biology. We're going to come back to that a lot. It turns out that these hydrogen bonds are very much causing the properties of water, that you have high freezing temperature, high boiling point. The energy of each hydrogen bond in water is roughly 2 kilocalories per mole. Can be slightly more in proteins. That's a number you should know, but we're going to come back and repeat it several times. And the reason why you have this in water, you might remember even from upper secondary school, is that the oxygen will effectively steal some electrons from the hydrogens, so that the hydrogens end up with a small partial charge, positive charge, and the oxygen with a small negative charge, and that means that that hydrogen is also happy to interact a bit with that oxygen. There is another molecule that's equally central, and that brings us more into the real biomolecular structure. That's DNA. You've probably seen this, well, I would be a bit worried if you haven't seen that molecule a dozen or 100 times by now. You see it all over popular science literature and everything. It's kind of the hallmark of modern science. It can, I think, yes, it's even embroidered into carpets. Virtually every single life science center in the world, including SciLife Lab, needs to have a model of DNA in their logotype. That tells you something about how important it is. Arguably, although I'm somewhat biased, arguably this is the structure of DNA could be considered the most important scientific discovery of the 20th century. At least if you have a bias towards biophysics. But that was not at all obvious when people determined the structure. Do you know what this is? In a way, it's not so bad. So chromosomes, the problem is science always makes sense retroactively. When we knew what we were finding, right? But when you make science, you usually start out with a mess and then try to make sense of it. So DNA was actually first isolated in the late 1800s. Somebody tried to find, then they were studying pus from infectious wounds and everything. And then they came up with some sort of other strange compound. And at the time, they had no idea what this was. But then eventually, in the first part of the 19th century, people were able to isolate this strange molecule that exists in all cells. And when you isolate the molecule, again, at the time, there were no obvious easy ways to determine what the molecule looks like. It's a salt or something. You can easily dry it out to get it into a salt, but what do you do from that? It's a salt. You can't put a salt in a microscope and see what it looks like, because the microscope is limited by the wavelength of light. The obvious way you could do with physics is, of course, if you turn the salt, if the salt is a proper crystal, you could shoot X-rays on it. And if it's now a sequence of crystals, then you have trillions and trillions of molecules, right? And then you're going to get a diffraction pattern from all these molecules. And then you can capture this diffraction pattern. Now, that is trivial for sodium chloride, 2 atoms. And You determine what is the length between two sodium atoms, what is the length between two chloride atoms, and what is the length between sodium and chloride. And from the patterns, you can see the type of unit cell you have in it. Easy. For DNA, in principle, the same thing works, but it's a slightly more complicated molecule. So this is a diffraction pattern from a DNA crystal, determined by a scientist who is not as famous as she should be, Rosalind Franklin. This photo, what the scribble there says is photo 52 on May 2nd, 1952. And that's all they had. What Rosalind and Franklin Gosselin did is that there's an amazing amount of things you can do just by approaching this systematically. So you see that there appears to be some sort of systematic difference between these bars, right? They're spaced roughly the same distance apart. And if you do your physics properly, and you're just going to have to trust me here, I would so not know this either. If you start to put out some length lines here, you can actually use those length lines to derive, well, first, this cross would correspond to that you're actually having some sort of helical shape in the structure. And these length lines will tell you something about the length scale, that there is some sort of repeat here with a few angstroms or nanometer. But that's all you can determine, because it's pretty darn low resolution. So you can't go from the left and tell what the structure is. That's impossible. What you then can do is that you can sit down pretty much with a molecular model and say, if I have a hypothesis how DNA would look like, I can test, would this hypothesis model lead to a diffraction pattern that would look like the one on the left? Then there are three possible answers. The answer might be, no, actually, in principle, there are only two. Your model could be incompatible with the data, or your model can be compatible with the data. It's impossible to say that the data proves that your model is right. And a lot of very famous people did this. What you have here on the right is a model by Linus Pauling, arguably one of the most famous scientists of the 20th century. So this is a helical model that uses the base of DNA, because we knew from a chemical composition, people knew what the bases in DNA looked like. It's just that we didn't know what the entire structure looked like. So this is a structure that looks like a spiral stair in the middle, with the backbone in the middle, and then you have the bases pointing out. This was published in Nature, 1952, I think. It's a completely incorrect structure, right? And like 50 years later, we can laugh at it. But anyway, that structure is compatible with the data. It's impossible. The data can't rule out that structure. So this is as beautiful as physics is with great models. Physics can also lead you wrong. It's not a stupid structure. It's one of the most world famous chemists and physicists in the world, but the structure was not right. We tend to sweep such things under the rug in the history of science, and I think that's really stupid. Because when we present science to you as students, we send the signal that every single prediction we ever made was right. And that's not the case. It's rather the opposite. 95% of the predictions we make are wrong. We're going to come back to after the break how we figured out that it was right. But there's a famous interview with Linus Pauling, not specifically based on that paper. That's 30 years later when it was famous with his Nobel Prize and everything. And there is this interviewer, Dave, I forgot what his name was. And I was, so Dr. Pauling, how does it come you have so many great ideas? And his answer is, well, Dave, it starts with having a lot of ideas. And the point is that is how science works. You need to have lots of ideas. You need to dare to make assumptions. And that's one of the things I want to teach you in this course. Don't be afraid of picking up pen and paper and start assuming things. And work on things and see where it leads. The worst thing that can happen is that you realize you get a beautiful model, and 2 pages later you're going to realize, oh shit, that model is completely compatible with the data. But that we're going to come back to after the break. It's 2 p.m., so let's meet here 1:15, and then I will continue. All right, I will get started again. So I'm going to tell what I told Burke and Lucy here in the break, that I know that you like structure a lot. So what I'm going to do for all of these lectures, I will try hard to stop sharply on time and not putting over more than 1 minute. That might mean that some lectures I'm going to run out of time, and then I just move the last 5 or 10 slides or whatever to the next lecture. So I'm never going to skip going through them. If we want to understand the DNA structure a bit, and this is something that I would like to ask you to do because it's hard to understand in 60 seconds, understand the different components of these nucleotides. You have the different bases that you've probably seen in popular science at least, A, G, C, and T, that then they're binding to a small sugar, which is also binding to a phosphate, and then these phosphates are linked into this long chain that was determined by Levene already in the early 1900s. There are a bunch of different ways you can look at the structure, but the whole point, the whole component here, each building block is called a nucleotide with T. And then you can have a number of phosphates there. You have the bases, which are the actual parts connecting to each other. And unfortunately, this was the only one I printed in your handout notes, but I think this might be a better picture, and you can find these online. So that the whole thing is called a nucleotide with T, and that consists of a phosphates, a sugar, and the sugar and the base together there is called the nucleoside, which is a nitrogenous base. Classical exam question to ask you to show. I will show you the molecule, and I will ask you to name what the different components are. These have been known for a long time, since the early 1900s. And during a lot of years when people spent time studying that, Erwin Chargaff found very interesting results, that it appears that no matter what organism you have different ratios of all these, but it appears that adenine and thymine always occur at roughly the same concentration, and guanine and cytosine roughly at the same concentration. What that concentration is, it varies, but they always occur in pairs. And that was the one key discovery that two other scientists used. So there are a number of problems with this structure, even without the Chargaff rules, that these outwards facing bases, they would need to make hydrogen bonds with water or something on the side. Yes?
I saw on the last slide that you have, they weren't exactly the same. What is that?
It's biology. Noisy. And you see, this is the problem for US physicists, because you expect to see this number with 14 decimals, right? 26.0 and 23.9, is that the same, Lucy? Yeah. That's how a biophysicist thinks. Because it's 5%, just a 5% measurement error? That's awesome.
To be honest, there should be an error associated.
Yes, it should be. But again, in biophysics, there should be plus minuses everywhere.
So this is due more to measurement errors rather than diseases?
But yes, but also there are so many things. You might have ended up with a bit of RNA in the sample, right? There are 110 things that could have gone wrong here. So that the key thing about focus on the first principle, the simplest possible explanation. Then the point here is to compare to 19 versus 30. That starts to be a significant difference. So the patterns. The problem with this structure is that you would have electrostatic charges. Those phosphates are negatively charged. So you're going to take lots of negative charges and put them right next to each other. That's going to be bad. There are going to be some steric clashes, that is, atoms will bump into each other if you try to build a model of this. So that it was not wrong, but it's not obvious that this model would be so great. And at this point, there were two other scientists that stepped in, and depending on the way you tell the story, you could argue that they pretty much stole Rosalind Franklin's discovery. There is probably some truth in that, because I don't think that they were entirely scientific honest about it. But I would also say the key difference is that they took somebody's data, but they spent more time thinking about it. And they came up with one amazing discovery. And there is a reason. I'm going to pass around this paper to you. I would suggest that you read this until tomorrow. It's a long paper, 2 pages in Nature in 1953. They came up with the reason why these must occur in pairs is that they must bond to each other. And suddenly you have an obvious explanation for the Chargaff rules. And then they started to build these molecular models you see there on the left, and it fit the structural data completely. And you see this point is that this explains the Chargaff rules. There was no obvious reason in the Linus Pauling model why we would have the Chargaff rules. And there's a beautiful formulation towards the end of this paper that you should remember. And that basically, it has not escaped, I don't remember the exact formula, but it has not escaped our attention that the proposed mechanism would also provide a mechanism for inheritance. Period. That's it. So that's basically their argument. This is how this is the genetic material of mankind. And that sentence, again, is indicative of the largest discovery in the 20th century. This structure also ends up being heavily stabilized by hydrogen bonds. It's the hydrogen bonds that's tying these pairs of bases together. And the hydrogen bonds, again, provides the molecular basis of this Chargaff's ratio, his rules. There are a ton of other stabilization features. If you look at that structure, you even have so-called pi-pi stacking. I'm not sure if you've seen that in a biophysical chemistry course or something, but you have aromatic rings, and these aromatic rings on the bases will interact with each other in very favorable ways when they're stacked this way. So DNA is a surprisingly stable molecule. it's also a slightly more complex molecule. Why would you have 4 bases? Being a physicist, you probably work with computers. How many states do you have in computers? 2, binary. So why would you have 4? That's more complicated. So if you only had 2 bases, and let's say that you have 8 positions, in 8 positions you can encode 256 different states. If you have 4 bases in 8 positions, we can record 65,536 different states. So it becomes a more compact storage when you allow slightly more different levels. This is incidentally what you use in modern flash storage too. You have more than 2 levels. You have multi-level storage. So it's a very efficient way of storing information in molecular structures. And there's a ton of information in DNA that we won't have time to go into. This is then, of course, long after the initial discovery, we've now been able to sequence whole genomes. And on the top you have an amoeba and the bottom mankind. There's a huge variation, diversity here in how large the genomes are. So a small genome could be just a few thousand base pairs. There are bacteria that are in the ballpark of 10,000 base pairs. Or at least 100,000. Mankind, how large are your genomes? That's a number you should know. 3 billion base pairs. Again, plus minus 10%. 3 billion base pairs. How many proteins does that correspond to? Because each such gene, well, the genome will encode for proteins, right? So how many proteins do you have in your bodies? Well, we did for a long time. We didn't know. We were guessing. We were guessing a lot. So when these first structural, and it's also, do you think that if you look at the previous slide, mankind certainly isn't the most advanced structure here. There are certainly some plants and everything that have 100 million years. As we were able to sequence the entire human genome, the sequencing was just the first step. The second step that have going on for 20 years now is trying to make sense out of these sequences. What proteins do they code for? There are some deep questions is that most of the protein in your cells doesn't code for anything at all. At least it doesn't appear to code for anything. So like 90% of your protein or of your DNA is kind of strange dark DNA that is important, but it doesn't appear to code for proteins directly. And that has been one of the long debates here, is that originally it was thought that just 1% of your genome coded for proteins. Today it might be 5 or 10%. I think, yes, we have a later paper on this where we'd argue that it's 8.2% coding for related to function at least. But something like 90% is just strange DNA. It's not useless. All organisms don't have this. In a bacterium, virtually 95% of the DNA will code for proteins. So we are different than bacteria here. And the general consensus today is that most of this DNA is regulatory. So you have pieces of DNA that can kind of shut off RNA, so that the DNA can control whether a protein should be expressed when you're young or when you're old. There's a classical example of fetal hemoglobin that has higher affinity to oxygen because the fetus need to be able to steal oxygen from the mother. Now, the second we are born, we no longer need that fetal hemoglobin, and then we shut off the gene. A bacterium can't do that, but a human can do. But this is not quite science fiction, but very much active research. So it's a bit of a mess. To come back to the question of how many proteins we have, that has been going down year by year. We thought at first was 50,000 to 100,000. The latest number pegs it at slightly below 20,000. So as fancy as you think you are not as particularly fancy. There are only 20,000 small molecules that determine everything about us, from the color of our eyes to the hair to their length, everything. If you compare it to something else, a Christmas tree, given the season, how many proteins does a Christmas tree have? Take a wild guess.
15,000.
Good guess, but wrong. 200,000. So we are roughly 1/10 as complex as a Christmas tree. We don't really know why. It likely has to do with evolutionary pressure. Well, I guess if you're a Christmas tree, it's not good to be beautiful. But the most extreme evolutionary pressure is bacteria. And that's likely why bacteria has had to focus on so few, maybe just 4,000 or 5,000 proteins, because they can't afford to carry around things that are not absolutely necessary. But all this evolutionary stuff we don't know. We're going to come back to that, the evolutionary pressure on biology and everything. There is another type of molecule called RNA, just to remember what DNA looked like. If you take that sugar, and in DNA we have an OH and an H group. In RNA you have two OH groups. It's a tiny difference, but this will completely alter its stability and how it behaves. So while DNA forms a double helix with this classical, RNA typically prefers to be a single strand. I'm not going to go into details exactly about how these reactions work. RNA, this AGCT bases we have in DNA, they're AGC and uracil instead of thymine in RNA. There is a molecular reason for that. I'll cover it in 30 seconds. And it has to do with that cytosine can actually chemically be degraded to uracil. Cytosine exists in both. It takes a while. If that were to happen if you had uracil all over the place in RNA, the problem is this would lead to errors all the time that would build up, and that would be very bad. So in DNA, that where we store information a long time, it's very important that they are different. In RNA, we typically just store information transitionary, so it doesn't really matter that much. And that's likely the evolutionary reason. It also means that RNA is not really stable. RNA will break down. You can't, if you work with RNA in the lab, you typically keep it on ice to make sure that it doesn't break down. What you get from these Neanderthal genomes and everything, that's always DNA. And there is a small famous story there that my uncle, he spent, in the 1960s, there were a lot of research going on to understanding how RNA breaks down and everything. There were lots of labs in the world, and he was sitting in Princeton working with it. And after a while, I think he got bored that the entire rest of the lab were working with RNA, and that someone said, well, can't I study DNA? And everything, but that's really stupid. Everybody knows that DNA is stable. So he started studying DNA and actually found out DNA is not necessarily stable. DNA breaks down too. And that's what got him the Nobel Prize in Sweden a few years ago. And this is the reason for a whole lot of cancers and everything. DNA too is degraded by orders of magnitude slower than RNA. It's Thomas Lindahl. I think we covered the structures already. You've seen the DNA. The RNA, we could plot it that way. Do you think that molecule would be stable? It would never look that way. So what RNA will do is that RNA will coil up. It's self-complementary, that this single chain will coil up into some sort of complex structures. And these are much floppier than DNA, and they exist in many more conformations. There are a bunch of different places where RNA occurs. We certainly have, if you look in the ribosome, which is kind of the protein factory in the cell, that's actually a mix of protein and RNA. So that the ribosomal RNA is the dark blue parts here. The dark blue is a small subunit, and the dark red is the large subunit. And I don't think we separate, no. It's slightly darker versus brighter shades that separates the DNA versus the protein part, but it's not that good. So the lighter colors here should be the protein, while the darker ones should be RNA. So this is a semi-horrible mix of RNA and protein. You don't even see the atoms here because it's so large. There is transfer RNA that is used when we're moving the small building blocks that's going to help us to build the proteins. Again, I'm going to come back to that in a second. And at some point now we're starting to have lots of different molecules. And the place where all of these molecules are used is when we build proteins. And I figure that's, in particular, since most of you are physicists, you might have seen this in upper secondary school, but you've probably forgotten most of it. So I'm going to take a couple of minutes here and, well, repeat roughly how proteins are created and how genetic material works, but introduce it in a slightly different way. This is something called the Central Dogma of Molecular Biology. The two books we have here, actually I can pass around the books if you want to have a look at, read them and see if you like either better. Both of them introduce this, but they introduce it in slightly different ways. So the Central Dogma of Molecular Biology can be formulated as sequence leads to structure, leads to function. And the sequence we have, that is what you have encoded in your genome, in the DNA. That's pure sequence information. Well, it is encoded by molecules, but its sequence is A, G, C, and T. This is converted through RNA to protein structure. And for a particular sequence of DNA, you will have a particular sequence of amino acids in a protein. And that will determine whether this is an antibody or an ion channel or hemoglobin or something else. given a particular structure, that will give it a particular function. For instance, hemoglobin carries oxygen. An antibody, well, that acts as an antibody. And a pump will maybe move ions across the membrane. But it's always sequence, DNA leads to structure, the protein leads to the function, what the protein does. If you want to change the function, we need to change the structure. And if you want to change the structure, that means we have to change the sequence. So sequence, structure, function. And the way this happens is that we need to start from DNA. And DNA is normally, when you see DNA, it always has this double helix on. But if it's a double helix, we kind of protect the bases on the inside, right? So to be able to read this, you need to start by splitting the DNA. And this whole sequence of events where you need to recognize the part of the DNA, a particular combination of bases where to start reading. Then we need to cut the DNA open. And we're going to need a small machine to do this. And remember what I said the first part of last lecture. If you need a small machine to do something, what would your first guess be? It's a protein. So we need some sort of protein doing this. This protein, of course, is also encoded for in DNA. So then we have a bit of a chicken and an egg problem, but let's not worry about that for now. So this is a protein called POLD or DNA polymerase. So this cuts up DNA, and it can then create copies of DNA. So you then have two strands. So first you're opening here, and now you have one DNA strand there and one DNA strand there. And that's how we can replicate the information. The other alternative is that, well, that's good if you want to create more DNA, but then we would live in a DNA world that we have more and more DNA. It's not useful unless we can somehow read it and move the information somewhere else. That we do with a slightly different protein. It's a process called transcription. Transcribe, read, something, right? So then we need to open the DNA in a small piece. And this is a molecule called RNA polymerase. So the polymerization process here is when we're stitching things together. That's why they're both called polymerases. And ACE is always an enzyme, a molecule helping you to do a process. But in this case, we're not copying it to a DNA strand. In this case, we're leaving the DNA as is because the DNA will close again, rewinding, but we're creating an RNA copy of the molecule. This is actually, it's not just a single protein. It's an entire class of proteins that are related to each other. This structure was finally determined high resolution in the early 2000s by Roger Kornberg at Stanford. It's quite fun because I was a postdoc in this department in a different lab at the time. And of course, around the year 2000, they were all working so hard in getting this structure. And it's a completely different way of looking at this while it's happening because people had no idea whether they would be succeeding. They had spent 20 years going after this structure. And nobody knew whether they would be successful. And of course, then they were. And coming back 2-3 years later, they were super famous for this. They were, in fact, so famous that they got the Nobel Prize for this structure, too. So at this point, we now have this red RNA chain. So now we have the genetic information, but we've copied it from DNA to an RNA chain. But that's still just a sequence of bases. And now it's very fragile. This is also fragile. This will break down spontaneously, as I mentioned, because RNA is not stable. There is a second process that does this. This RNA moves into a molecule called the ribosome. This is an old slide. It's a super old picture. It's old. It's 2005, 4 years after I got my PhD. Again, in the early 2000s, we had no idea what the ribosome looked like. So this ribosome, as I mentioned, is a complicated structure, both with pieces of RNA and protein and everything. So this takes the messenger RNA coming in, the small red chain there, and then had this other piece of RNA, transfer RNA, that is kind of small carriers. And each such carrier has bound one amino acid. And then you just have a sequence of these carriers. We're getting more and more and more of them. And then what the ribosome does is that it then stitches all these amino acids together into a long chain, and that's how we create the protein. And there were a number of groups involved here, but in particular Tom Stites, Peter Moore, and Venki Ramakrishnan. I got the Nobel Prize in 2009 for this, with X-ray structures. So we're talking about fairly modern science here. Again, it's only 15 years ago these structures were not available at all. So what you effectively do here is that you always have triplets of bases. Each triplet of base will uniquely code for a particular amino acid. And it was originally Francis Crick who discovered that. And this concept was we call the genetic code that I'm sure you've heard about, but you might not remember it. This table you need to know by heart. No, you destroyed my joke. No, you don't know it by heart. Nobody. Actually, the sad part is that there are molecular biologists that know it by heart, but you're physicists. You're not going to know it by heart. Completely useless knowledge to know by heart. So how many different ways, if you have 4 bases and you stitch 3 of them together, how many different combinations can you have? That would require the molecular biologist to bring out the calculator, but you're physicists, so I expect you to be slightly better at that part. 4 to the power of 3, right? So 4 by 4 by 4. That's 64. 64 combinations. Do you know from upper secondary school how many amino acids there are? 20. That's not the same as 64. So they lied to you. There are 44 more. No. So there are a couple of caveats here. First, we need to know when to stop. You need to know where to start, but that can actually be redundant with an amino acid. But the point, we need to decide when to no longer code. And there are actually 3 of these combinations that are stop codons. So when you see a stop codon, we assume this is the end of a protein. We're happy. But even then, there are 61 remaining. That does not mean that there are 41 amino acids you don't know of. So there's a built-in redundancy in the genetic code here. And this is also a classical exam question. If you look at the amino acids, I'm not sure whether you can see the table here or in your log. Do you see that some amino acids are more common than others, right? Some amino acids, there are at least 4, 5, 6 different combinations that code for arginine, while the poor tryptophan is pretty much just one combination, right? So which amino acid do you expect to see more of in cells? Tryptophan or arginine? Yes. So this, and how, if you just were to measure it, would you expect to see? Do you think that you see 6 times more arginine than tryptophan?
Yes.
You do. Again, within the normal 10 or 15% variation. This is what explains the relative abundance. That is, i.e., how much we have of each amino acid. Not evolution or anything. It's all based on physics, mathematics, combinatorics, whatever you call it. Simple code. These are the building blocks that we can combine them in different ways, but we can't change the building blocks. Yes?
Do you mean that it's just random? Shouldn't the DNA be able to adapt to do some of these more common than others?
That's probably what DNA has done over 4.3 billion years.
But I mean, if it's 6 times more, just because it's 6 times more likely.
So that now, of course, we're talking about in general, right? In your entire genome. Not in a specific protein might. There are, of course, some specific proteins that might not have any arginines, membrane proteins, for instance. But these are the general. Let's compare this with Lego. The relative distribution of all Lego pieces that are being produced by Lego Inc. That's one thing. The Lego pieces I use for a particular, when I'm building a particular toy, that's a different matter. But those are the building blocks that are available, and the relative abundance of them in nature is determined by the genetic code. And again, Francis Crick discovery. So to go from there, once we know what these building blocks are, right? And this was discovered in the 1950s. The other obvious thing is that can't we just determine the structures of these proteins with X-ray crystallography? Because we know that there should be a unique structure. Again, sequence leads to structure, leads to function. If the structure is not unique, it's not going to have a unique function. So there has to be a well-defined structure that only depends on these building blocks. So if I just know the sequence of the DNA, I can translate that using this code to the sequence of the amino acids in the protein. And then I should be able to determine, if I have 100 amino acids, I just need to find a way to determine what the protein looks like. And that seems like such an obvious problem. And people started that. They actually started that in parallel with the discoveries of DNA. And one of the big heroes in this field was Max Perutz. It took them 22 years to determine the structure of hemoglobin. Again, in hindsight, it was worth it. Can you imagine starting a project now that you would finish when you're my age? And you're not sure it's going to work. Talk about pouring your passion into something, right? Again, today it's simple because we know that there have been 5,000 other people in the world that have determined structures. Easy for me to do one. But being the first one, that is what real science is about. The other thing that we are so horribly spoiled with our computers, this is how they were determining their structure. They were sitting with rulers and measuring the distances between every single atom and building them up manually. There were other, in particular, hemoglobin. There's another protein called myoglobin that was determined in parallel. We will come back to what those differences are. Since then, there have been a number of structures determined. They got the Nobel Prize for the first structures, of course. Ion channels, these structures, when I was a PhD student, the first ion channel structure appeared by Rod McKinnon. I remember there was a small talk out at Karolinska when the guy came over and presented his structure, and people weren't really, I and some other people went because I was a bit interested in membrane proteins, and it was so embarrassing because nobody wanted to go out and have dinner with the guy, so that the host there convinced me and another colleague that, can't you join us for the pizzeria? It's a bit embarrassing otherwise. So we were 4 people sitting around the table. 3 years later, of course, he got the Nobel Prize, and now there tend to be slightly more people who want to meet him when he's in Stockholm. These are the structures that you have in, actually this is not human shape, but these are more similar to the proteins that exist in humans, where you actually have ion channels controlled by voltage. Aquaporins, these are the water channels that determines how much water goes in and out of your cells. Peter Ager shared the Nobel Prize with Rod McKinnon in, actually not 2001, 2003. GPCRs, the ones I talk about, Brian Kobilka, also Stanford, got the Nobel Prize for those structures in 2012. And the way virtually all of these structures have been determined is through X-ray crystallography. And that's what I said. Just take your proteins, grind it into a powder. So powder just means you will still have crystals, but they're going to be randomly oriented crystals. And then you put them in a very small droplet. We're talking about a microgram of crystal or something. And then you use a very large facility like this Max 4 down in Lund, a synchrotron. So you're using you're having electrons rotate around in the ring. And then when you use magnets to force the electrons to accelerate, so you're changing the direction, then they're going to cause synchrotron radiation lights. So you get very high intensity X-ray pulses. And then you use that and collect the scattered rays. That is so not film today, of course. You have computer CCD sensors. And then you're able to determine that type of patterns. It's slightly faster than 22 years nowadays because we have computers to help us. But then we determine what is the electron density corresponding to that pattern, and then you use computers to build a molecular model. But this can easily be a project that takes a few years. Just getting a crystal can take a year, if you can get it at all. But still, for a very long time, the safest way to get a Nobel Prize in chemistry was to determine the structure of a new important building block in your cells. And there are probably still more in the pipeline. It's a bit sad. When I first gave this course, I had all these GPCRs. You could say, oh, this is likely a future Nobel Prize. And that was so cool when I could say the next course round, and this was awarded the Nobel Prize last year. So a few years ago, there are other ways to determine structures, but historically nothing has come close to X-ray crystallography. And this is the beautiful way when things change in science. They change much faster in biophysics than in traditional physics. There is another way called cryo-electron microscopy. I have a paper on this that I'm going to pass around too, if you're interested. 3 pages, but the first page is just an image. This is a 2015 paper in Nature. So cryo-electron microscopy builds on the fact that you've heard about or seen electron microscope pictures, right? And the whole idea is replace the light with electrons. If you just accelerate electrons to a few 100,000 electron volts, they will have very short wavelengths, and then you can image with electrons. That is amazing in material science. You can literally see almost individual atoms and everything. So that way you can see structure that are as tiny as you want. The problem with that is that you only see that if you throw a lot of energy. You need to throw tons of electrons on this. And these proteins are fragile. So if you start to throw lots of electrons on a protein, it will break. And then you don't see the structure anymore. So you need to have very, very low doses of electrons. And you need very sensitive detectors. The only problem if you're shining electrons on those detectors, the detector breaks. Most things don't like 500,000 electron volt electrons being shot at them. So for a year, everybody was joking about calling these field blobology and everything because you got these faint outlines roughly of the shape of the structure. But then through a sequence of events, suddenly there was a generation of new better detectors, the microscope were better. So during 2 or 3 years, this was literally like turning a page. So suddenly the method was good enough. So today, the hottest technique in the field, and we have 2 high-end microscopes of this out at SciLife Lab, is that instead of collecting diffraction patterns, you have a very, very thin film of frozen protein sample, maybe just 100 nanometers thick or something. And then we're literally using a microscope. Well, it's an electron, and then we have a real sensor here, and then we're collecting images about these proteins from, say, 100,000 different directions. Now you have slice-through images of your protein from 100,000 random directions. And that you throw in a computer and tell the computer, please fix this. And the amazing thing that it works. We're going to, well, no, I don't think I'm going to cover it that much, but it's an example, right, how modern experiments would not be possible without computers. And you can imagine the amount of physics and math that have gone into these reconstruction algorithms. So modern biophysics is in many ways more computational and mathematical than it is experimental. So there are some very cool proteins in that. Up on the left here, that was the structure. It's a TRPV1 protein. I'm not even going to go through the details what it does. But up on the left there, that gray outline was how well you know the structures in the blobology days. We couldn't tell anything. And then Yifan Sheng, a few years ago, they were able to get 3.4 angstrom structures. Do you see all the detail? You can see the individual helices and everything. And we're going to go through these components and structures on Friday. We can find the binding sites. We can understand what these molecules do. And in particular, these molecules, they're actually pain and heat receptors. So if you're eating chili peppers, this molecule, capsaicin, binds to this channel and causes the channel to open. And that is how you sense pain or heat. And of course, there are hundreds of structures like this. Virtually every issue of Nature now has 1 or 2 high-impact papers with a new structure determined by cryo-electron microscopy. So the unifying factor with all these things is that by having the right sequence of DNA or a particular sequence, we can build a chain. Actually, in some cases, in this place is 4 different chains, but that's a complication we can go back to later. We can build a chain of amino acids. This particular sequence of amino acids will magically, for now, form some sort of structure. And what I'm going to argue, but that I can't prove to you yet, that process is based entirely on physics. And you might think that that's obvious, but it's not at all obvious. When people started studying this 60, 70 years ago, they went, oh, maybe the cell has some sort of very special machinery for building all these proteins. But the fascinating thing that the entire structure, the way that this protein always finds the same state, is based entirely on physics, which is remarkably cool. But once it has that particular structure, it creates the specific binding site here that causes us to bind that molecule. The way the entire molecule looks, it's what gives us the properties. If it's binding that molecule in the right place, the binding of that molecule will change the state the entire ion channel would like to be in. So suddenly, when we have bound this small capsaicin molecule, suddenly it's better for the molecule to move over to another state where it's open. Then, about say a minute later, but probably a fraction of a second later, this molecule unbinds. And now it's better for the molecule to move back to the closed state again. So these are stupid molecules. They're literally just molecules, right? But because they can visit, they're large, they're way more complex than, say, water or carbon dioxide. They can exist in multiple different states. They will literally move. And that's a complication, because how do you see those motions in X-ray crystallography? We can't, right? You could imagine determining 2 structures. You could, first we determine a structure of just the protein, and then I can take my crystal, but before I make the crystal, I can soak the crystal. I can add lots of this molecule, and I hope that this molecule will be bound everywhere in the crystal. And if I'm really lucky, I might hope maybe I can see one structure with the molecule and one without it. But how do we actually see the process when it's moving? So historically, the answer is you can't. And this is where the modeling comes in, understanding the differences between different states, and where virtually all modern pharmacology and everything is after, because you can imagine that if all these molecules steer and control your nervous system, what if we could deliberately steer them? What if we could create the molecule that's a better anesthetic or something? But that will literally require us to be able to go in and fiddle around with the process a little bit. So what I'm going to talk about on Friday, the reason for this diversity is this polypeptide sequence. And polypeptide, I spoke about amino acids. There are lots of different names here, and some of these things will just slip out of my mouth. A peptide string is just a sequence of amino acids. And the reason why I call it the polypeptide, I'm going to come back to this, is that these amino acids they polymerize through something called peptide bonds. So each amino acids end with a COO minus group, and then the next amino acid typically starts with an NH2 plus group. Sorry, NH3 plus. If they move together, and I think we have, yes, I have an example of this. So sorry, I have these two amino acids. If they move together, they can release a water and instead form a larger molecule that sits together. So I have one amino acid there and one amino acid there. And if this happens again and again and again, this is what's happened in the ribosome. Then I create a very long chain of amino acids. So the point is that there are only 20 amino acids, but if I now have a string of, say, 10 of these, there are now 20 to the power of 10 different combinations. And that starts to be fairly large numbers. So by connecting them in specific strings, I get much larger diversity. But just the string is not enough. The string just means that I have a string of amino acids. What tells different proteins apart is that there's R groups that I had on the other slide. Again, that's literally what makes one amino acid different from the other. So you have a tyrosine there in the middle, you have an alanine there, and you have a glycine there. So different side chains, the ones in red here, will cause a diversity. And we will come back to this too in the course, that this whole polymerization process is fairly simple. You deal with polymers in everyday life. Plastic is just polymers. The ethylene is a small molecule derived from ethanol. The ethylene, you might have heard of polyethylene, PE plastics, every single shopping bag. But those are fairly boring, right? But the reason why they're boring is that every single monomer is the same. It's just billions and trillions of copies of the same monomer. For amino acids, you can have different monomers. And because they're different, we get much greater specificity and much cooler molecules. But fundamentally, a whole lot of things are going to be kind of, from the physics point of view, part of this is similar to plastic. When it comes to sequencing, it was Fred Sanger in 1952 who argued that proteins have a unique sequence. He managed to sequence it. There is a paper on Sanger's first paper on sequencing that he got the Nobel Prize for in Canvas, if you want to read it. It's a bit more chemical, so I didn't include it in the things I hand out, and I'm not going to cover it in detail. But given how, at SciLife Lab, we sequenced roughly the equivalent of 1 human genome per day nowadays. The first human genome was a worldwide endeavor that took 10 years, and now we do it in 1 day at one site. And Fred Sanger sequenced insulin, and it probably took him 3 or 4 years. So there's no other method that creates more biological information today than sequencing. It's exceptionally powerful. These are old structures, beginning of the 1900s. So the fascinating thing, all this research on the proteins on the one hand, protein sequence on the second, DNA structure and DNA sequence, funny, they all happen in parallel. And it wasn't really until the mid-50s or early 1960s that we realized that all these things stick together. Good, Jesus, for once I'm on time. So I mentioned that the DNA structure was arguably the world's greatest discovery in the 20th century. Do you remember from the slides when the Watson and Crick paper showed up? When was it published? You have the paper in front of you. 52. When did they get the Nobel Prize? It took over a decade. So how on earth could it be that it took a decade to award the greatest invention, well, arguably the greatest discovery of the century, a Nobel Prize? So that at the time we didn't know, because the Nobel Committee, their deliberations are secret. They're secret for 50 years. So about a decade ago, this was lifted, 6 years ago, it was lifted and people could say, so they were not even nominated until the early 1960s. Nobody understood how important the DNA structure would be. And there's this fallacy, we tend to look at the future, but we think the future will look like the past, right? Science is obvious when you look at the rearview mirror, but in 1958, nobody could imagine that DNA would be important. It was just another small molecule that could be useful. Small fun paper, they determined the structure of a specific salt, might or might not be important. And beware of this, that the challenge with research, it's hard to predict, it's hard to make predictions, in particular about the future. And today it's completely absurd that nobody bothered nominating them. It was Sir Lawrence Bragg who nominated them originally. So the summary today, you need to understand a little bit of why protein structure and function is important, how they relate to each other, how do we determine structures today, understand the hydrogen bonds. We're going to come back to that. I didn't talk that much about it here, but the hydrogen bonds will be important later on. We will go back on Friday and talk about fundamental physical interactions and how they give rise to the hydrogen bonds. You need to understand DNA and RNA structure and function, because even if we're just going to work with proteins, this is where proteins are coming from. And you so need to understand the central dogma of molecular biology. And that leads to one question here. So what was the central dogma?
DNA to RNA to protein.
So those are in the molecules, but if you think in terms of information and sequence to structure to function, right? So do the arrows always go in that order? Sequence to structure. Do the arrows ever go the opposite direction? Does the sequence ever depend on the function? In principle, no. Not when we talk about the central dogma. But there is a very important arrow that everybody is aware of this arrow, but we don't point it out. But you need to think about it. There is an arrow that goes this way. What is that? Evolution, yes. So this is basically what Charles Darwin found. And that's another one, because that's 200 years before we knew of any of these structures, right? So evolution literally means survival of the fittest. So if you have sequences that are more or less randomly created, if that leads to structure that have a good function in some sense, this structure will have these individuals will have an advantage in evolution. They will likely get more offspring. If they have more offspring, those random deviations will start to become more common in the sequence. And then you have an amplification out there, which is also quite fun, because biology obeys the laws of physics, but it's not static in the sense that we can change the sequences, and biology does change the sequences. Reading-wise, for the first lecture here, I've actually found chapters both in Nordlund and Finkelstein that cover this reasonably well. Finkelstein doesn't cover DNA and RNA at all. But again, the first approximation, and you can find for the general stuff, you can find a ton of information online. I'm going to update the following lectures too already today and tomorrow, so that whenever possible, I will have reading instructions for both books. On Friday, we're going to go deeper into the sequence structure function relationship, and we're going to talk in a small protein like this, how do all these things interact? Why do they interact? And why does that lead to stabilization of certain functions? Before that, Magnus started to do this last year. I think it's a good idea. If you want to be successful in this book, this is what we talked about in the course evaluations. Last year, what did they recommend you to do? Actually, I think this is from 2 years ago. They thought that attending class helped a lot in understanding the book contents. If you prefer to see this online, be my guest, but that's up to you. They also strongly recommended starting reading and do hand-in tasks in the beginning. Don't see the hand-in tasks just because they have a deadline on February 3rd. I promise you that we're not going to grade you down because you completed earlier. And you will have a chance to interact way more with us. Somebody else who liked the lectures. The other part, this is a famous quote that I actually, there was some other quote by Ralph Waldo Emerson. Nothing great was ever achieved without enthusiasm. I am frequently super busy, but when I am teaching, you have my attention and enthusiasm. I love to teach, although my time is limited. If you want to be successful, not just in this course, research, life, and anything, what you do it with a full heart. And I will start sharply on time. I will end sharply on time. I will be available over email even. And again, while we give this course, you have my attention. I will respond quickly unless I'm on a flight. But that also means I don't want you to show up 10 or 15 minutes late. Things happen. In that case, go in quietly and sit at the end. But so then take responsibility for your teaching, and I will make sure that I deliver to 110%. And I think the same goes for Lucy and Burke. The other thing that I will do, sorry, now I use that last minute, study questions. Every lecture there will be a bunch of study questions. If you know those study questions, you're going to pass the multiple choice questions and the exam, I promise. The multiple choice questions will be taken from these. So that's if you know these, you know that lecture well. And that's it for today. See you on Friday.